Okay, so it's time now to talk about the primary visual cortex, also known as Brodmann area 17, striate cortex, or V1. So here it is. Here's the brain viewed from the side with area 17 or V1 marked on in green here. Here's the brain, or half of the brain, viewed from the middle side. And you can see that a lot of V1 is hidden away in the calcarine fissure, or the calcarine sulcus more accurately. It's also known as Brodmann area 17 because of Corbinian Brodmann, whom you might have come across. He was a 19th century German neurologist, and in what I think is a truly heroic effort, he defined 52 distinct cortical areas purely based on the cellular architecture. So he noticed that the 6 layers of cortex differed in their thickness and in the type of cells across the brain, and he was able to distinguish, as I say, these 52 areas long before anybody had any idea what they did or whether these differences were significant, or was he just making them up, but they really map on incredibly well to the areas that we now recognise. So here are Brodmann's areas, and area 17 is shown here in yellow, and you can see it agrees really well with the diagram of V1 that we saw earlier. So I believe it was Brodmann who originally described the 6 layers of cortex, and in visual cortex, or primary visual cortex, layer 4 is especially prominent, and it actually appears as a broad stripe, and that's why V1 is also known as striate cortex or striped cortex. So nowadays we subdivide layer 4 into A, B and C, and layer 4C is further subdivided into 4C alpha and 4C beta on the basis of different connectivities. So that stripe is known as the stria genari, or the stripe of Genari. So it's actually made up of a band of myelinated axons that project into layer 4B from layer 4C alpha. Another feature of primary visual cortex is that it has what are called CO blobs, so little localized regions which contain a high concentration of cytochrome oxidase and thus a high concentration of mitochondria. You can see that very beautifully in this diagram here from a review on the circuitry of V1 and V2, which again I'd recommend to you. So in V1 at the bottom here you can see these little patches, and those are the CO blobs, and in V2 you get this quite a different pattern. So you have these thin and thick stripes of high CO regions. Now the projection from the lateral geniculate nucleus of the thalamus to primary visual cortex is both layer specific and retinotopic, and I'm going to be talking about what both of those terms mean in the next few slides. So we saw the different layers of the LGN, which is represented here. So we have the 2 magnocellular layers, contralateral and ipsilateral, from the contralateral eye, from the ipsilateral eye, and the 4 parvocellular layers, again ipsi contra, and then the coniocellular layers in between that are shown in green. So the 4 parvocellular layers project predominantly to layer 4C beta in V1. From the magnocellular layers, the ipsi and the contra, these both project to layer 4C alpha in V1. And the coniocellular layers, they project to blobs in V1, layer 3. So these cytochrome oxidase blobs that we saw in the previous slide, they are receiving input especially from the coniocellular layers. The coniocellular layers also project directly to another brain area called MT, but since I'm not talking about MT at the moment, we'll get to that later. We've seen that the LGN cells are monocular, so each LGN neuron is receiving input from only one eye. Those axons then travel to V1, where they terminate in layer 4, and as a result you can see these beautiful ocular dominance columns like zebra stripes in V1. So here's an experiment where one eye has been injected with a radioactive substance. That substance has then got taken up and transmitted back to LGN and even further back to V1, and you can then see these radioactive axons coming into V1 from the injected eye. and you can see there are those bright regions from, let's say, the left eye, and these dark regions in between from the right eye. And you can see that rather than being sort of arranged at random, information from the two eyes is grouped in this rather beautiful striped pattern. Now that's just in the input layer, in layer 4 of V1, and elsewhere in V1 information is brought together from both eyes.